So this is the standard off-the-shelf panel that called the pan-cancer panel that Pillar has. And before we went into getting our own custom panel, we wanted to make sure that the panel works. So we took this off-the-shelf panel, and most of the data I'm going to show you is from this panel. 54 genes, I believe, in here, covers a whole lot of genes related to solid tumors, some heme genes as well. Doesn't include some of the genes that I mentioned in our custom panel, but it's a great panel. A little bit more about this panel, and I think Manfred spoke a little bit about this. Sample type, you can use genomic DNA, FFPE, with the 1% variant detection is what Pilla says. I come from New York State. It's really hard for me to go to 1% and say confidently it can do 1% with the kind of coverage we have. So I like to say about 3%. DNA input, 5 to 60 nanograms is recommended. We have gone as low as 2 nanograms, and it works really well. Amplicon size, you see over there, the on-target rate is excellent over here, 98 to 99%. The coverage uniformity is excellent, and those are all details that one can see in the software. It can detect 1 to 2% of variant allele fraction, and that is what, I'll show you some data with that. So this is the workflow for the ONCO reveal panel. Manfred showed you it's 8 hours. Of course, I would say, yeah, it can be 8 hours, but generally not 8 hours. It's a little more than 8 hours. You know, techs don't work an 8-hour day from start to finish with all these things. So we normally start in the afternoon and finish it the next day. So I'd say it's about 1.5 days. Just the points to bring out over here. So you have your target amplification. I've given you hands-on time and the total time for each step. These are steps that are really, come really right one after another and are very simple to follow in the same tube. Finally goes on from primer digestion to indexing to library purification on Ampere beads and normalization like anything else, and then finally pooling and going on to the MySeq. After it goes on to the MySeq, it runs overnight on the MySeq, and then the FASTQ file is uploaded directly to the website for the PIVAT software. We have also used our own bioinformatic pipeline for this, a very simple bioinformatic pipeline that we use for the 48 gene panel, and it works fine. So from there, we take the VCF file from the PIVAT software. We have our own knowledge base that we put together. The two of them merge together to give you a report. This report is then cut and pasted directly into COPATH for us, and it goes alongside the pathology report for clinicians. Okay, so this is the specifications for each run. We run 22 samples at a time. This is what we have tried on a single V2 flow cell. The neoplastic content of what we've done is anywhere between 20% and 80%. We have now tried 10%, and that has worked, but I do not have the data for that over here. Our input, I told you, was from 2 to 50 nanogram, and the library QC, as you can see down there by the bioanalyzer, is pretty sharp. Shows you really neat peaks over there. Remember, this is room temperature, just 2 small PCRs, single tube amplification. For that, it looks really good.